All right, hello to my friends who are joining us via recording. Today is April 7th. We are wrapping up lesson number 11 about the brain, and if we have some time, we're going to try to do a little bit of Unit 3 review. So it looks like some of the things that we're interested in talking about are what's going on when a patient's in a coma. So we'll definitely take some time to talk about that. We're interested in the types of blood vessels in the brain, the reticular activating system. So let me make myself a little list on my paper here. We want to talk about RAS, coma, blood vessels. I know I saw a comment about sensory areas, the types, primary and association, so we'll see. Maybe between this stuff we might have enough material just to stick with lesson 11 today. So let me look at my pictures here and we'll see what comes earliest in your outline, and we'll start with that material. By the way, I liked this particular meme that I found this morning, that the brain is the most important organ you have according to the brain. What might Dr. Aulis say the most important organ or organ system would be? What's my favorite one? Yeah, digestive system. Although I think probably, I think an argument could be made that the brain is probably more important because it controls the digestive system, to be fair. But yep, the digestive system is still my favorite. So let's check out, looks like the first thing I hit in my pictures here is the reticular activating system. So the first thing in the order of events from what I have is the reticular activating system. Oh, let me mute. Help me out. What page on our packet is the reticular activating system on? Can someone help me out? Looks like I'm here in maybe page 7. Yeah, page 7 it sounds like. So you're looking at a picture from page 7. The big thing, first thing I'll mention here with the reticular activating system, let's put its name on our screen here. I'll put it in black. We're looking at what's called the reticular activating system, or I like to just call it RAS when we're talking about it. You will see on the homework assignment and you will see on the exam that I'll give you the name. Oh, that's on page 9. Okay, well, find whatever page we're on. I don't know what page it is. Reticular activating system, or RAS. I will give you both names when we are going through and we're doing an exam or doing an assignment. So first thing I'll mention about RAS is that it's not like when we talk about other structures in the brain and it's really easy to say where it is. Let's do a little bit of review for ourselves because this will help us out with lab stuff coming up here later in the week. When I talk about the brain, what's the name of this part of the brain back here? What's the name of this little thing that's down here at the bottom? Can we tell? Yeah, several of us are chiming in. This one back here is called the cerebellum. So the cerebellum, the best way that you can identify it is it's down low. It's really folded. It's got lots of bumps here on the outside. So I've got the cerebellum down here. This really big part of my brain that I see up here, the largest part of your brain, the part that yesterday we said is what makes you human. What do I call this big whole thing up here, this whole big part? Yeah, a couple of us have chimed in. This big part of your brain, the part that has all of the lobes in it, this big part is called the cerebrum. So we have the big cerebrum that, remember, has its gyri, its bumps, and its sulci, the little indentations. And then we've got this little part here in the back called the cerebellum. The cerebellum and the cerebrum look really similar to each other. So they have a lot of a similar anatomy, but the physiology of them, the cerebrum, because it's so much bigger, does a whole lot more different things. And we talked about a lot of those things yesterday during our class time. We didn't talk a lot about the region of the brain called the brainstem in our class last time. So this area right here is called the brainstem. One of the most noticeable parts of the brainstem is this big part that sticks out in the front. This big part that sticks out in the front, its name means that it's a bridge. Does anyone remember what I'd call this part right here? Yeah, several of us are chiming in. This big sticking out part, probably the easiest part to identify on the brainstem, is called the pons. The pons is the bridge. The pons, the best way to find it is it's directly across from the cerebellum that we identified. So we've got the pons that sticks out right here. Down below it, there's another little bump. This is my last brainstem structure, and this structure is the one that actually attaches to the spinal cord. So this one down here is called the Medulla Oblongata. I'll type its name for you because that one is not English at all. Medulla Oblongata. That's the lowest brainstem structure that I see down here. That second bump that you see on the brainstem. And remember from our, yeah, the medulla for short, absolutely. Remember from our discussion yesterday, because we did talk a little bit about the brainstem. The last part of the brainstem is this top part up here. And the top part up here is called the midbrain. And really the only thing that you care about in lab for the midbrain is the fact that it has these little bumps on the back. Let me give you another not English word at all. The name of those little bumps back there, as a reminder for us, they're called the Corpora Quadrigemina. The Corpora Quadrigemina, that's these 4 bumps that I see here in the backside. Right now I just see 2 of them because my brain is cut in half, but there are 4 of them here on the backside of the midbrain. So all you care about for lab purposes is that the midbrain is where I find these bumps. In lecture, I'm not even going to ask you anything about the midbrain because it's really just the location where those bumps are found. So that's my brainstem. We talked about the cerebellum. We mentioned the cerebrum. Remember then from last week when we were doing our brain ID stuff that there's this middle part of the brain, and it looks kind of funky on my picture here because I've added all these arrows for the reticular activating system. But here in the middle, does anyone remember the big part that's inside here? I called it the duck face in our lab discussion last week. Does anyone remember what this big part here in the middle of the brain was called? What was this area here? Yeah, Kathleen chimed in for us here. This big area here in the middle is called the thalamus. And remember, the way I kind of talked about it last week was it's the duck face. So I've got this big area called the thalamus inside the very middle of the brain, the duck face of the brain. Hanging down below it, the bill of my duck down here, this region down below is called the hypothalamus because hypo means below. And then our regions here in the back are called the epithalamus, although the one that we care about the most for lecture purposes is this little one I'm going to circle right here. See this little feather kind of that looks like it's sticking off of that region there. Does anyone remember what I call that little structure? We used it interchangeably with the thalamus. Yeah, Ariel's right. That is my pineal gland. Pineal gland. That's the only thing. The epithalamus is more than just the pineal gland, but what we really care about in lecture and really in lab is the pineal gland. So that's the little pineal gland, some of those feathers on the back side of the duck head there. So a quick anatomy review for us, because I know we've got lecture exam this week, right? But coming up fast is the lab exam too. So can't be forgetting about that one too. So use the pictures that you can in the lecture packets and the lab packets to help you review for both of those exams. Okay, so we started this discussion by saying that we're looking at this picture or using this picture for something called the Reticular Activating System. The reason that we went through and talked about all those other recognizable parts of the brain is because when I talk about the Reticular Activating System or when I talk about RAS, there's not one structure that I can pin on a model and say this is the Reticular Activating System. So the good news is you won't ever have to find this in lab because it's really just groups of neurons. A lot of them live, as you can see in my picture here, in this brainstem region right here. So most of them are clustered in the brainstem, but then they have effects. Like we can see all these little purple arrows that you see coming out of the thalamus region here. All of those little purple arrows are places where the reticular activating system kind of spreads out to. The big idea with the RAS is that it's not one structure. It's kind of a bunch of neurons that go all over the place in the brain. For my friends who have worked on or read a little bit about RAS, help me out. What might you say some of the functions of RAS are? What are some of the things you remember reading about that the reticular activating system does for you? Okay, consciousness, it's what wakes you up, keeps you alert. I love it. Yeah, these are good ideas here. So the reticular activating system, this is what we use to stay activated, if you will, or to stay alert. That's kind of a good way to remember. Alert, or in the medical community, we'll say conscious and oriented. So if you've ever read really technical doctor's notes, a lot of times when they're checking you in, they'll ask you, remind me of your name, tell me your birthday, tell me where you are or why you're here. They're kind of doing like a consciousness check for you to make sure that you're aware of your surroundings. So we check to see that the reticular activating system is activated to keep your brain conscious, to make you able to respond to the environment, to keep you alert. Somebody mentioned that the reticular activating system is what wakes you up. Absolutely. So in the morning, what's going to get you to wake up is when your reticular activating system turns on. The way that the reticular activating system works, you can see all these little purple arrows here. All of these little purple arrows are representing messages that we're sending out to the brain. So all of these little messages, the way that your lesson talked about them, think about them kind of like a submarine that is in the very depths of the ocean floor. There's nothing to see around it. So the way that it's going to get around and not bump into something is it's going to send out little sonar pings, little sound waves, and listen for those pings to come back. So the little pings come back for a submarine, to help the submarine know where it is. The little pings go out into all of these places of my cerebrum up here in normal circumstances to make those places awake, or at least to the technical word that we might use for what it does is it primes the cerebrum. Or if we don't want to use the word priming, we could say that it gets it ready. Gets it ready to receive messages. See, I know there's a lot of chatter going on in the chat about when it's on, when it's off. Times when the reticular activating system isn't working are times when you can't receive other messages, or times when around you, you aren't perceiving what's happening. So for example, when you're totally passed out sleeping, man, my 3-year-old is giving up naps. We're gonna remember the quarantine as the time that she potty trained herself, which is awesome, but also the time when she officially decided she was done with naps. So what that means is there are times where she's just gonna crash, and she'll be sitting in the living room watching TV and she'll totally pass out, and she's not gonna hear anything and she's not going to see anything. She's not processing any of what's going on around her. That's because her cerebrum is not activated anymore. Her cerebrum isn't alert anymore. So the goal of the reticular activating system is to keep the cerebrum awake, to keep it alert to perceive those things. So I know a couple of us already mentioned this in the chat, but let me ask it formally here. When a person is sleeping, when they're just sleeping, I mean, they're kind of dead to the world, right? But they're passed out. What's going on with the reticular activating system when you go to sleep? When you take a nap, right? A lot of us are saying we haven't given up on our naps, right? But man, 3-year-olds, they think that they're just the worst thing ever. Yeah, so a lot of us have talked about it. When we are sleeping, we turn the reticular activating system off. So basically think that we put it in, it has an on-off switch. When you're sleeping, you turn it off. We deactivate it, it's no longer working. We can turn it back on, and we do turn it back on. We have to be able to wake up. One of the things that I'm going to make you guys help me out with, I'm not just going to give you that information. There are several different things that can flip the on-off switch for the reticular activating system. And they're labeled on the picture, and you were supposed to identify them when you were going through the lesson. What are some of those things that can help us to turn the reticular activating system on when I've turned it off? Or what are some things that could activate it for me? Okay. So I'm seeing light, Definitely that's something. I'm seeing sound. Carrie mentioned bright light therapy. That relates maybe more strongly to another structure, but bright light therapy would definitely turn it back on. Yeah, some of us are mentioning touch sensations or pain. For those of you that haven't had a chance to work on this part of the lesson, it's not that your classmates are just mind readers and they can tell exactly what I'm looking for. They're actually just using the parts that we see on our picture here. So check out right here, number 1. This picture shows me how RAS works, how the reticular activating system works. So the way that it works, step 1, is that sensory neurons collect information. So the kinds of information that they can collect are the kinds of things that we have listed here. So one of the kinds of information that the reticular activating system can collect is visual information. So this is the fact that in the morning when the sun comes out, it starts to get brighter outside. Visual impulses are something that feeds into the reticular activating system and tells you that it's time to wake up. A lot of us mentioned auditory, sound things. So this is why we set alarms on our phones that make sound, right? Because the auditory information that feeds into the system as well. When the music goes off or the jingle goes off, we hear that and that wakes up because it feeds into the reticular activating system. Oh, and here's a good review from yesterday's office hours. Notice how it mentions here general sensory information. General sensory information. So remember that we had special sensory information, and that was some of the things that we talked about, like vision or like hearing. General sensory information was kind of all the other sensations. So it doesn't include taste, it doesn't include smells, it doesn't include balance information, but things like touch sensations or temperature sensations, pain, the kind of stuff that we listed over here, that's considered general sensory information. A lot of us have jumped ahead of me because it was a question on your assignment, right? There's one very important kind of information that does not feed into this system. What's the very important kind of information that does not feed into the reticular activating system? Exactly. Yep. So we got the technical name for it, and then we got the easier name for it. So the technical name for the kind of information that does not feed into the reticular activating system is olfactory information. Olfactory information does not feed into it. That's the technical name. The name for it is smell information. So information related to what you're smelling does not feed directly into the reticular activating system. That means that it's not going to be the primary way that we activate this system. Now, Eileen brings up an interesting point. She says that smell information does wake her up. Here's what my thought would be for you with the smell information, Eileen. We're going to talk this more in our last lesson together when we're talking about the process of smell. When we talk about smell is something that's called a chemical sense. What you're actually detecting is chemicals in what you're smelling. My guess would be that when you feel like a smell is what's waking you up, what's actually waking you up is some kind of general sensation related to the chemicals that you're smelling. So a lot of times students ask me about, well, what about like smelling salts, right? So you use smelling salts and sometimes it looks like it wakes somebody up. Well, when the salts and the chemicals inside of them come up and touch the stuff inside your nose, that registers as a chemical that perhaps is sick, and it registers kind of as a pain sensation, or we might be registering some of the other sensations. But we're not necessarily specifically registering smell, at least not in the way that we would normally consider smell information being processed. So in general, for the intents and purposes of what we're studying to get us ready for the upcoming exam, we're going to say that the kind of information that doesn't process in the system is smell information. We can't do smell with this system. We can see things. That's why flipping on the light is going to wake you up. We can hear things. That's why setting an alarm makes you wake things up. Things like touch sensations. So if someone comes and touches you, if the temperature suddenly gets really hot or something is really painful, those are the kinds of things activate the reticular activating system. Did I cut out for a minute there? It looked like I might have. Can we hear me now? Let me ask that a little bit. Okay. Okay, so let me look at my chat really fast. Yeah, so some of us are getting to that critical thinking question. I know I saw. So when you can't. So earlier somebody asked. When you can't hear your alarm clock, is your reticular activating system malfunctioning? It's possible, or we need to make our sound sensations a little bit louder. When you're really, really tired, right? Sometimes it takes a lot of stimuli to turn this system back on. So it's possible that it's malfunctioning and that's why you couldn't hear your alarm, or it's just possible that like all of us, right? We need to sleep a little bit more. I'm in that boat with you guys. Let me see. Parker asked if it's possible for the reticular activating system to be hyperactive. I don't know if we would say that it's particularly hyperactive, potentially, but what can happen is it can just not want to turn off. So this is what happens when you're having a really hard time falling asleep, right? So this was me Sunday night, which was awesome because Sunday night I stayed up super late writing your exam. And so then I go and I lay down in my bed and I'm like laying in my bed saying, curse you, reticular activating system. Like I just want to sleep. So one of the things that feeds into the reticular activating system over here is the visual impulses. Let's do a little, this is half soapboxy at you here, but one of the kinds of visual impulses that really feeds into the reticular activating system is light that is in the blue or purple wavelengths. So blue light or purple light, the kind of light, remember, that people rag on about our phones and our computer screens. So this kind of light, blue and purple light, normally, like if we weren't in the kind of society that we're in right now where we've got screens everywhere, way back before screens, the only time your body would be exposed to blue and purple light was in sunlight. So if you're being exposed to blue and purple light at midnight or 1 o'clock in the morning, your reticular activating system thinks it's still sunny outside. So then you go and you lay down in a dark room and your reticular activating system says, but it's light outside. Like, why are we shutting off? So the best kind of lights to keep the reticular activating system going are lights that have that blue-purple undertone to them. So maybe it'd be possible that RAS is overactive, but what I think might be more likely is that there's stimuli that are going into it, like blue and purple light, or like sound sensations, like some of these different general senses that are feeding into it and are just not letting it turn off. So that would be my thought, hyperactive RAS thing. Let's see. So one of the critical thinking questions that I asked you guys about then was to apply this to why we need smoke detectors. And a couple of us have put some thoughts into the chat here. Let's approach it from a slightly different perspective. When I talk about a smoke detector, what are some of the things that smoke detectors do? When a smoke detector goes off, what does it do? Loud sounds, yes. Loud, angry sounds, yeah. And Ashley mentioned something else that can do too. So makes loud noises. Mine used to yell at me. It would say, fire, fire. And then it would go, And it's like, oh, man. Because the only time it ever went off is when I'm cooking pizza, right? So, like, you burn your pizza, and we're telling the world that I need to get out of my house. So first thing smoke detectors do, they make really loud noises. Second thing that a lot of us have noted now is that they flash a light, right? So they flashing light. Okay, let's think about this in terms of the reticular activating system. Things like a loud noise Is that the kind of thing that feeds into the reticular activating system? Do noises feed into the activating system? They do. Yeah, auditory impulses. That's one of the big ones. That's what we use to wake us up, right? Normally, is sound sensations. And flashing lights, is that the kind of input that goes into that system as well? Do we do light impulses? Yeah, we see it over here. Visual impulses also feed into the system. So I've got two kinds of stimuli that feed into the reticular activating system. The goal of a smoke detector is to wake you up, right? That's the reason that we have them, and that's the kind of the way that the question was phrased. People die because they don't wake up when there's a fire in their house. Because think about some of the other kinds of sensations that feed into the system. Things like temperature sensations. If you are waiting for the temperature in your house or in your room to get high enough to wake you up, the fire is probably already in your room, right? Or it's too close for you to get out. Exactly, it may be too late. Same thing with pain. If you are literally on fire and that's what's waking you up, it's too late to get outside. If we were awake, the first thing that would help us know our house is on fire, what's the first way we would know that our house is on fire if we were awake? What would be the first way? Well, even potentially before our alarm. How do you know when there's a fire if we're in a different place of the house? Okay, so some of us are saying we might see smoke, but with smoke, where I was trying to go with that is we would also probably smell the smoke. If we were outside hiking, for example, and there was a fire that was a couple miles away from us, probably the only sensation we'd know that there's a fire going on over there is a sense of smell. So the sense of smell is theoretically going to be the first way that you would know there's a fire. But if there is a fire in your house and there's smoke around, does the sense of smell feed into the reticular activating system? Would that wake you up, that sense of smell? It wouldn't. Yeah. The first thing you'd probably notice if you were awake and your house was on fire is you would start to smell the smoke. That would give you plenty of time to get out of your house. You'd be safe. Except the sense of smell does not feed into the reticular activating system. So the reason that smoke detectors keep you safe is they provide you with a noise and they provide you with a flashing light. And the goal of both of those things is to activate this system to get your brain to wake up. So it gives you the kinds of information that you need to wake up before it's too late. I kind of like that terminology that someone mentioned. Before it's too late to get out of the house because it's super hot or because you're in pain, let's wake you up with things that would wake you up. Now, the question that got us into the reticular activating system, though, is what's going on when a patient is in a coma. So help me out, for those of us that have worked on this lesson. What did we say was happening for patients who are in a coma related to this system? What's going on with the reticular activating system when you're in a coma? Yeah, it's not working, it's damaged. Yeah, the wet that we used on the assignment. So when you're in a coma, your reticular activating system is damaged. Or when we want to kind of explain what that damage looks like, are we permanently in the on position or permanently in the off position? What happens when we're in a coma? Yeah, we are permanently, or at least as long as the coma lasts, we're permanently off. Permanently off. So what that means for you is the kind of stimuli that used to turn on this system won't turn on this system. And remember that the job of this system was to send out pings or to wake up the rest of the cerebrum. If I can't send out those pings to wake up the rest of the cerebrum won't be working. So when we talk about a patient in a coma, the biggest thing that they've lost is what we said that RAS does for us. The biggest thing that a patient in a coma has lost is they're no longer alert, they're no longer conscious, or they're not aware and able to respond to their environment. So when a patient is in a coma, the thing that causes them to go into a coma is the reticular activating system gets damaged, or we've turned it off in a way that's deeper than just them sleeping. And so Ashley asked a great question about a medically induced coma. We do have the ability in medicine to put a patient into a coma. This is especially helpful. For example, let's talk about the current pandemic, right? In patients that need to go on a ventilator, a lot of times we will put them into a medically induced coma because the idea of having a tube running down your throat is not great for your cerebrum and for a lot of your reflexes. So yes, we will use a medically induced coma. I like the idea of using the word altered for that. That's a good way to think about it. Altered in the sense that we have turned it off on purpose. It's not that it's damaged. When we take those meds away, it should activate itself and come back on. But yeah, it's a great way to think about it. Eileen mentioned burn patients as well. Absolutely. Times when there is incredible pain or things like brain swelling or like I mentioned, having being intubated, those would be times that we might want to turn off the system on purpose to block some of those normal functions. So Rose is getting more specific with us here because the question on the homework assignment asks you what is and is not working. So let me pull up a whiteboard and I'm gonna put this out to the class. So the question on your homework assignment, let's see, when a patient is in a coma, and remember we had this qualifier, but they're still breathing on their own, I don't have to breathe for them, what is and is not working? Okay, so we're going to make two columns on our chart here for what is working and what is not working. The first thing on our what is not working list, we literally just talked about. What would be the first thing I'm going to say is not working because I'm in a coma? Yeah, RAS. Exactly, RAS. Okay, so reticular activating system is not working. That, by definition, is what puts me into a coma. When we talk about my patient still being able to breathe, this ties in a little bit to what we talked about in class yesterday or during our office hours yesterday. What part or parts of your brain helps to regulate the process of breathing? What keeps you breathing? Okay, yep, one of the places that keeps us breathing is that medulla oblongata. The medulla oblongata, that's one of the places. The medulla oblongata works together with, yeah, Ariel mentioned for us, it works together with the pons. The way I know that these two structures are still working for me is because we had this qualifier, my patient is still breathing. Now, if this patient has to go on a ventilator, if their lungs stop working on their own, we would have to shift over the medulla and the pons over here too, because the way I know they're working is my patient's breathing. When we're breathing, we had a place, we talked about this yesterday, the Pontine Respiratory Center. We had a place called the Medullary Rhythmicity Center. These two places keep you breathing. So my patient's breathing, I've got these things working. I don't have the notes in front of me. Let me pull them up really fast because I know I gave you guys a list of structures to sort. So let me pull that out really fast. Updated the brain. Let's see. Okay, so let's go through our list. And yeah, it was on page 12, like somebody told us. Two of the things that were on our list were the precentral gyrus and the postcentral gyrus. Let's start with reminding ourselves, I'm going to put this not on a list because we need to sort it. The precentral gyrus, we talked about this a lot yesterday. The precentral gyrus has something inside of it. What do I find in the precentral gyrus of the brain? What's the structure that lives there? Does anyone happen to remember what we said lived or is found in the precentral gyrus? Yes, yeah. Kathleen and Rosa are chiming in for me. That motor map, right? This is the location of the motor homunculus. Really weird picture that we looked at. When I talk about the motor homunculus, is the motor homunculus about collecting information, or is it about sending directions? What does the motor one do? Collect information or send information? Yeah, that's my sender, right? That sends information. Specifically, the motor homunculus sends directions to skeletal muscle. Skeletal muscle, the one kind of muscle you do control. So one of the structures that we want to figure out if it is or is not working is the precentral gyrus. That's the location where this motor map lives. When a person's in a coma, are they consciously controlling their skeletal muscles anymore? Are they doing movements that they want to? Yeah, they're not. Yeah, when a person's in a coma, they don't move a whole lot. So we're going to add our precentral gyrus to our list of what's not working, because that patient is not up walking around, they're not brushing their hair, they're not answering their phone, they're in a coma, they're not consciously moving. So precentral gyrus makes our list of what's not working. Next thing on our list that I asked you to think about is the postcentral gyrus. What do I find in the postcentral gyrus? If the precentral gyrus has the motor homunculus, what does the postcentral gyrus have inside of it? Yeah, a couple of us are chiming in. It's the exact same thing, it's just the sensory version. So the sensory homunculus, that's found in the postcentral gyrus. And this is the one when I ask you if we're sending information or if we're collecting information. This collects information. Collects information. Yeah, collects sensory information. So when we talk about the postcentral gyrus, I would be, well, let me just see where we stand right now. As a class, we think the postcentral gyrus is or is not working. What do we think? What's your votes? Is or is not working? Yeah, so we're kind of split 50-50, and one of us says not sure. I think a case could be made either way. Here's what I will say about the postcentral gyrus. The postcentral gyrus, it helps us to collect information and to kind of feel things. What I will say with that, yeah, and Rosa kind of honed in on here's where I was going with it. With the postcentral gyrus is found in the cerebrum. When we think about collecting this information and feeling these sensations, I would really say that for a person's postcentral gyrus or their sensory homunculus to be working, they have to be conscious. So conscious awareness of touch sensations, of temperature sensations, of those kind of things. When a patient is in a coma, is that patient conscious? Are patients in a coma conscious? No. Yeah, that's the definition of a coma. They're not conscious. So I would say that patient who's in a coma, I would say central is not working. That would be my inclination with it. I know that cases would be made. Some patients out of a coma, they stay now. Sorry, am I back now? Hopefully. It's tight. See if the audio comes back kind of. I'm telling you guys, it's going to be once a day, drops coming. Well, look at it. Poor. Be back now. Back. Right, my computer is not absolutely. Computers are mad. Okay, can we keep again? Barely. Yes. Okay. I'm telling you guys, it's going to be like once a day. We were doing so good. Well, like our first week of sessions, no problems. And then I think like everybody must be using Blackboard to meet. So hopefully that was our once a day. We'll see if it kicks me all the way out again, like it did yesterday. I hope not. Um, Okay, are we back? My internet completely died. Sorry. Okay. I'm not sure what happened on that. So I might do actually is I might just stay on mic to see if that helps my internet work better. So I'm going to try to just stay on mic with you. If I go in and out again, and I'm out again. Man. Okay, hopefully I am back again. Okay. Like I was saying, I'm just gonna try to stay on mic. On my end, it looks real scary. It says my connection's bad, so I think it must be something with my internet. I guess everyone's going back to online school today, and they're jamming up our waves. Well, I can't see what you said before then, so hopefully it wasn't too mean, right? I can't see what it was, so you're good. Okay. Where we were before my internet completely died was we were trying to figure out where to put post-central gyrus. What I would say for the sake of our studying is the place we should put post-central gyrus is on what's not working. Because the last thing that I chatted with you about before my computer died is that for the post-central gyrus to work, we need to be conscious. And when someone is in a coma, they are not conscious. So I would say that the post-central gyrus is something that's not working. They're not consciously aware of the touch sensations in the way that you would be when you are awake, or in the way that normally a touch sensation could wake you up. A patient who's in a coma, those touch sensations wouldn't wake them up. So I would say we should put post-central gyrus over here on what's not working. Let's look at our list of other structures. We needed to know if the cerebellum was working, the basal nuclei. So let's add those to our list down here. When we talked about the cerebellum, what did we say that the cerebellum does for the body? What is kind of the jobs of the cerebellum? Does anyone remember? Yeah, so one of the big ones was balance. Yep. What was the one that related to movement? Does anyone remember the one that related to movement with the cerebellum? Yeah, it helps to smooth out our movement. Yeah, so balance, smooth movements, that kind of thing, coordination, all of those are functions that the cerebellum helps out with. We said earlier that patients who are in a coma, their precentral gyrus is not working. So we're not doing any of these kind of movements that need to be smoothed out. We don't have things that need to be coordinated. Based on that, I would say that the cerebellum is not working as well. These patients are not managing their sense of balance. Again, a lot of this comes back to the fact that they're not conscious. So anything that kind of requires you to be conscious, probably not working in a patient who's in a coma. What did we say yesterday that the basal nuclei did for our body? We talked about a couple of conditions when the basal nuclei aren't working. Yeah, so we're thinking the basal nuclei aren't working in a coma patient. Yep, what they do? Yeah, they're controlling that unwanted movement, or they're regulating our movements. So blocking unwanted movements. That's kind of big picture what the basal nuclei do for us. So we talked about the two conditions. We talked about Parkinson's disease, where a patient has constant tremors. Tremors are movements that occur for a really long time. Or we talked about Tourette's, which are really short movements that in Tourette syndrome, they have what are called tics. So we are not blocking these kind of movements. Well, we're not doing, let's go take a step back here. Blocking unwanted movements is what the basal nuclei does. A patient is not doing any movements at all. So with the basal nuclei, we don't even have any wanted movements that we're trying to complete. Because of that, we don't really need the basal nuclei working as well. Their job is kind of irrelevant because we're not moving our skeletal muscles. So when a patient's in a coma, they don't typically move. Kathleen asked if they're moving. They don't typically move. And the big idea with the basal nuclei is they're blocking any, we've used the word static before with kind of messages that are going around in the brain. The basal nuclei are supposed to be there to stop static, except there is no static because none of the cerebrum's really working. So I would argue that a patient that's in a coma, their basal nuclei are also not working as well. Let's go back to our list here. We already talked about the reticular activating system. So we need to mention the limbic system and the pineal gland. Let's add those to our list down here. So when I talk about the limbic system of the brain, what do we generally say is going on with the limbic system? What does the limbic system do? Yeah, so one part of it does fear. That's definitely part of it. Rage, yep. So fear and rage, those are things that are done by what's called the amygdaloid body. That's one part of it. Yeah, big picture, when I talk about all the limbic system structures, it really comes down to emotions. This is the way that your lesson described it, the emotional brain of the body. So the limbic system is the emotional brain. This is emotional responses to smells. This is fear and rage. This is memories. Let me put it to the class. Do we think that is or is not working? What do we think? The emotional brain. No, yeah, maybe. This would be one that probably is in the no category because, again, the biggest thing is the fact that you're not conscious. Limbic system, add it over here. But here's what I'll say, and this has been kind of coming up in some of our comments as we've been making our list over here. I would say that it might be possible for the limbic system to possibly be working simply because you hear about people that come out of a coma and they say, I remember hearing people talking about this. Or someone mentioned reading to patients in a coma. Sometimes patients come out of a coma and say that I made a memory of something happening. It's not, we definitely have to make the caveat if we're saying that it's working, it's definitely not working at full capacity the way that it would be if a patient was conscious. So the limbic system, we could go either way. It probably isn't working because you're not conscious. So we kind of need to be conscious to really feel emotions. But the fact that some people can remember what happened when they're in a coma, that's a function of one of the structures in the limbic system. What's the limbic system structure that helps you to form memories? Does anyone remember what that one was called? The limbic system structure for memories. Yes, it was the hippo, right? The hippocampus. Yep. So the hippocampus is the place in your brain that we store memories. And memories are very closely related to our emotions. So if you're feeling emotional, that helps you remember things better. So we could argue either way. Probably overall, we'd say the limbic system is not working. But if you wake up from a coma and you remember stuff, that's your hippocampus in action there. Next thing that was on our list of structures we wanted to put places is the pineal gland. The pineal gland. What would we say the job of the pineal gland is? What does the pineal gland do? Helps with sleep. Yep, particularly by, that sleep hormone. So the big thing with the pineal gland that we want to know is that the pineal gland makes melatonin. Pineal gland, melatonin. When we talk about making melatonin, it would be possible for your body, because we're in an environment where we could regulate the lights, turn the lights off at night, turn them on during the day. It would be possible that the pineal gland might actually still be working. So I'm going to put the pineal gland over here on the is working side of things, because the pineal gland makes melatonin in response to light. So as long as they're turning lights on and off around you, that light can get through your eyelids. Your pineal gland may be on track. Now, when we're talking about a patient in a coma, because they're in a coma and not just sleeping, if this was a patient that was just sleeping way too much all the time, that would probably be a problem with the pineal gland. Because remember, the melatonin that the pineal gland makes kind of helps to turn off the reticular activating system, helps to calm it down. But this is not a problem when a patient's in a coma. It's not just that I've got too much melatonin and that's putting them to sleep. It's like I can't get the reticular activating system to turn back on. So we could go either way on the pineal gland. We're going to assume that the doctors and nurses are doing a good job taking care of you and they're turning the lights on and off, and we are making melatonin regularly. We'll say that one might still be working. We have left the thalamus. That's the only structure that we have left on our list here. When we talked about the thalamus, what were some of the things that we say that the thalamus does for the brain? And I'm going to have to rely on my friends. We didn't talk about this last time. So for my friends who've read and worked on the lesson, what are some of the things the thalamus does? What are some words you might use for it? That one's a little bit trickier. We can flip through our notes to find it. Okay, yep, one of the first things that we said about the thalamus is that it's a relay center. Relay center meaning that information goes through here. So the thalamus is a relay center in the sense that all of your sensory information goes to it. And the thalamus, I like the way that Eileen puts it, sorts info. First thing the thalamus does is you get a bunch of information in your brain and the thalamus sends it out to the places it needs to go. So first thing, thalamus is a relay center to help you sort that information. The second thing that the thalamus does is it can actually edit the information that's going to and from places in the brain. Does anyone remember the silly example that I gave you guys in the lesson about the kind of information the thalamus just edits out? What's something you're doing right now that you're not consciously aware of until you start thinking about it? Yeah, the fact that you're wearing clothes, right? Hopefully most of us are wearing clothes, not that anyone would ever know, right? If you're not, but the fact that you don't always feel the fact that you're wearing pants. The example I always give when I'm in class with students is I always mention shoes, but like when we're doing online school, like no one's wearing shoes, right? So I couldn't say that you don't notice that you're wearing shoes because most of you aren't wearing shoes, right? Let's be real, I'm not even wearing shoes, like we'll be honest here. So I am wearing clothes, however. Slippers, there you go. Yeah, so the fact that you don't always feel your slippers, the fact that you don't always feel the clothes that you're wearing, that's the kind of information that your thalamus just filters out for you, because you don't need to constantly feel the fact that you're wearing clothes until you start thinking about it and asking yourself, am I wearing clothes? And then you feel it again. So the thalamus helps to prevent sensory overload where you're just feeling everything, because if we didn't filter out some of it, we would feel everything. When it comes to whether or not the thalamus is working, what do we think on that one? Do we think the thalamus is working? It is or is not working? What do we think? Yeah, a lot of us are saying no, and I would tend to agree on that as well, that the thalamus is probably not working. The reason I would say the thalamus isn't working is because, remember, when you're in a coma, really your entire cerebrum's not working. So my thing that sorts information to different places in the cerebrum, it can't do its job either, because none of those places in the cerebrum are like accepting messages. So I would say that the thalamus probably is not working as well. Yeah, well, actually, it's not that it's not receiving input, because someone could be holding your hand and that kind of touch sensation would go to the thalamus, but the thalamus doesn't really have anywhere to send it per se. So it doesn't have anywhere for that information to go for it to be used. But it is still, since your body still exists, it probably is still getting a little bit of input. Rosa asked, isn't the pineal gland in the cerebrum? The pineal gland is in the cerebrum, but it doesn't have anything to do with consciousness. So you don't consciously regulate your pineal gland. Your pineal gland regulates itself. It's kind of the same way when we were talking over here about the medulla oblongata and the pons, how these structures are still working when a patient is in a coma, it's because you don't consciously regulate the part of your medulla oblongata. Well, you don't consciously regulate anything with the medulla or the pons. So it really kind of comes down to conscious regulation. So maybe it's better for me to say things that have to do with consciousness aren't working. Things that don't have to do with consciousness are working. So the pineal gland doesn't have to do with consciousness. That would probably be an argument that the limbic system probably shouldn't be over here, right? But the medulla oblongata, the pons, the pineal gland, since you don't regulate those at all, they're probably still working in the background. Yeah, I like the idea of comparing it to our muscles, right? Voluntary and involuntary. That's a good comparison for us. So yeah, things that you have conscious involvement in, since we're in a coma, consciousness is gone. We probably wouldn't have those. Things that just regulate themselves, things that keep you alive, like the brainstem, those things would definitely still be working. So yeah, so the limbic system, Carrie, the reason that we put it here with a little asterisk is that sometimes people wake up from a coma and say that they remember things. Some people wake up from a coma and say that they remember touch sensations, for example. So some of these things are a little bit fluid, but the big things we know for sure are still working are the medulla oblongata and the pons, probably also the pineal gland. Limbic system probably could go on the other side. Big picture when you're studying this, the goal of this activity. So here, let's put it here in the middle. Big picture. What we would probably say about this activity, the goal of this activity was I wanted you to think about the functions. So what are the functions of each of these structures? And when we're thinking about the functions of the structures, we also want to know what is or is not influenced by you being conscious. Basically, that was the goal of this activity, which is why things like the limbic system are a little bit dicey. Things like the pineal gland maybe is a little bit dicey, but the goal that I wanted you to get, what I wanted you to learn out of this, is I wanted you to know what do these things do. If a patient is in a coma, which of these activities are they or aren't they doing? Eileen asked if it will be on the exam. It's not going to be on the exam in this exact form. Or what I will say is on the exam, I might ask you, especially for these guys here, the medulla oblongata and the pons, they definitely keep plugging through when a patient's in a coma. Something like the reticular activating system, that causes a patient to be in a coma. And something like the precentral gyrus, you've got to be conscious to control your skeletal muscles. So the very obvious ones, especially not the ones with a star, right? I'm not going to ask you one that could go either way. But the really big ones, like the medulla oblongata and the pons, the reticular activating system, precentral gyrus, you could expect to see those on the exam. So again, big picture, what I want you to get out of this activity. If you know what each of these things do, that's half the puzzle. If you have an idea of whether or not consciousness would play a role in it, that's the other half of the puzzle. So big picture, that's what I want you to know from this coma activity. Any last minute questions about these big picture questions? Because again, we could debate some of where it could be up for debate, absolutely. But big picture, make sure you know what these guys do. Make sure you know whether it's conscious or unconscious. That's kind of where we were going with things. All right, well, give me a thumbs up or an emoji. Give me a dancing emoji if I get a dancing emoji today. If you're feeling okay, if you're ready to move on. We've hit 2 of our 4 topics so far. We have done RAS and coma. Awesome. I got some dancing going on. Perfect. Oh, here. Yes, it wasn't until most of you were gone. Let me give you our daily penguin here because I'll forget until later. Little penguin over here. For those of you that are new to Dr. Allis's penguins, Dr. Allis doesn't draw a lot of things, but one of the things she likes to draw is little penguins, cartoon penguins. And the kind of penguins that Dr. Allis likes to draw, let me finish our penguin for the day. The kind of penguin I like to draw are called macaroni penguins. So that's the reason why when I first drew a penguin, someone was freaking out about whatever these little things, whatever they thought these were up here. These are little feathers. Macaroni penguins have feathers on their heads. So here's your daily penguin that I drew for you. So got to get in that daily penguin today while most of you are still here. So there's our daily penguin. All right. Next thing on my list, we want to talk a little bit. Ooh, there went the whiteboard. Sorry. I hope you enjoyed the penguin while it lasted. We want to talk about blood vessels. So let me go and find our blood vessels. Do we want to start, help me in the chat, do we want to start with the ones that do or the ones that do not leak? Which ones do we want to start with? Leaky or not leaky? The ones that do leak? Okay, we can start there. That's a good place to start. That'll help us out too when we're talking about cerebrospinal fluid. So let's start with these. I'm going to be a little mean here, and I'm going to ask you if you can type for me. Does anyone remember the name of these leaky blood vessels? The blood vessels in the brain that do leak. What are these leaky ones called? Yeah, the choroid ones. Yep, so the name of the blood vessels that I'm seeing in this picture here, these blood vessels are called the choroid plexus. The choroid plexus. And that is going to be a lab word for us this week as well, the choroid plexus. When we talk about the choroid plexus in the brain, these are very leaky, very leaky. Most of the blood vessels in the brain are not leaky, but these are the exception. They're very leaky. But they leak on purpose. The choroid blood vessels, the choroid plexus blood vessels, they leak because they're making something that my brain needs. What does my brain need that the choroid plexus makes? Yes, several of us are chiming in with the abbreviation. That's all we gotta type, right? Not the whole name. Technically, they make what's called, I'll write it for you, cerebrospinal fluid. They make CSF, cerebrospinal fluid. Way back in Unit 1, when we talked about the different types of fluids in the body, CSF was one of them. Cerebrospinal fluid was one of them. So cerebrospinal fluid has a couple of different jobs. One of the things it does is it makes your brain feel lighter. So your brain is literally suspended in fluid in your cranial cavity. That is good news for you, makes it lighter. It also kind of acts as a cushion around the brain. So cerebrospinal fluid starts off as the fluid that's inside my blood vessels, inside my choroid plexuses. Hey, remind me, here, this will help us review for the final exam, the name of the extracellular fluid found inside blood vessels. What kind of fluid is inside here? When the fluid's inside here, what's it called inside here? Plasma, absolutely. Okay, so remember that with fluids, we kind of got like a circle of life thing going on here. So in the circle of life of cerebrospinal fluid, the fluid starts as plasma inside the blood vessel. Most of the blood vessels in the brain are going to hold onto their plasma. They're not going to leak out that plasma. But when we get to these places called choroid plexuses that are very leaky, they will leak out this plasma, and this plasma then is going to get filtered, or it's going to get worked on by these cells that I see right here. So these cells that I see right here, we probably want to underline, highlight, star them. These guys are called ependymal cells. The ependymal cells filter the plasma that comes out of these blood vessels. Ideally, they're making sure nothing bad gets from the bloodstream into the brain. Ideally, that's the goal. Once the ependymal cells are done working on this fluid, they release it here into the brain tissue. So on this side, we're into an area called the ventricles, fluid-filled spaces in the brain tissue. So we release it into these ventricle spaces where it's going to float around, where it has, it becomes, fluid becomes our CSF. Remember with fluids, way back to Unit 1, it has everything to do with where it's located. So I guess I shouldn't have gone away from there. CSF is found inside ventricles. If plasma is inside a blood vessel, CSF is inside a ventricle. So my fluid leaks out of the choroid plexus. My ependymal cells filter it for me. They spit it out into these areas called ventricles. Now those ventricles are filled with cerebrospinal fluid. And if you notice on my picture over here, we have labeled that some of the stuff that cerebrospinal fluid has, it has glucose and it has oxygen. Why do we like this glucose thing here? What's glucose good for? Yeah, when we're talking about our cells, any kind of cell in the body, this is how I make my energy. Glucose is my food. So one of the reasons I need to make cerebrospinal fluid is to get food to my brain, to various places. I also need that oxygen. Remember that oxygen, here's a tie-in to our muscles chapter here. If I'm going to make a whole lot of ATP, if I'm going to make a whole lot of energy, the best way for me to do that is to use oxygen. I know I'm going to stretch you a little bit. When we talk about the type of muscle tissue that uses oxygen to make their energy, does anyone remember the name of the process that an oxidative muscle tissue uses to make energy? Yeah, Carrie chimed in for me. I use oxygen in what's called the electron transport chain. Your neurons like to use the electron transport to make their energy. So I've got to get oxygen up there so they can use the electron transport chain. And I've got to get glucose up there as well. Because remember, neurons, the only thing neurons can eat is glucose. So the reason I make cerebrospinal fluid is to get nutrients and to get oxygen up to my brain. My choroid plexus are these really leaky blood vessels. They're going to let all kinds of things out. Kind of what I like to talk about with students about choroid plexus is if you get a brain infection, if there's something in your brain that shouldn't be there, it probably started here. Because most of the blood vessels in your brain would not let things get out. If the choroid plexus gets a little too leaky, if these ependymal cells don't quite filter good enough, that would be the source of a brain infection. Yeah, so I mentioned something called meningitis. Yes, when you have a brain infection, one of the ways that usually shows up is a condition called meningitis. We talked about last week in lab the meninges. Remember that meninges are connective tissue. So the connective tissue that wraps around the brain, meningitis is when this, oops, can't spell it. Meningitis, still can't spell it, whatever. And that's just terrible. Is when these guys get swollen. And when they get swollen, they put pressure on the brain, and that's bad news for you. So meningitis is when your meninges, your connective tissue around the brain, gets all swollen and angry. It's really bad when it puts pressure on your brainstem. Remind me again, what does the brainstem do for your body? What's the job of the brainstem? Big picture, the brainstem, things like the pons and the medulla. Have a couple of competing options. Help me out here. I promise it's not a trick question. The job of the brainstem in your body. Ariel mentioned it, Rosa, Robbie, exactly. The brainstem keeps you alive. So it's not great when the connective tissue around your cerebrum, your thinking parts of your brain, gets swollen, but it's deadly when I put pressure on brainstem. That's why someone will die from meningitis, because there's too much pressure on that part that keeps you alive. So if you get meningitis, if you've got a virus or bacteria that makes this connective tissue swell, probably the place in your brain or how it got up to your brain was probably because of these leaky blood vessels here, choroid plexus. Now, I'll mention with the choroid plexus that the choroid plexuses, what makes cerebrospinal fluid, are found in various places in the brain. This picture, you should note in your lecture packet that this will be really helpful when you're reviewing for the lab. So in this week's lab packet, we ask you to talk about the different ventricles and the different structures that carry cerebrospinal fluid through the body. This picture shows you all of those structures. So some of the things you're going to see in that lab packet that we have to be able to describe or identify in a picture, things called arachnoid granulations, things called the lateral ventricles, the fourth ventricle. See, there's my third. You're going to see a lot of these words pop up in the lab packet this week. So this is a good picture. Honestly, I kind of mostly included it for the sake of lab for you here. But let's talk about it. Choroid plexuses, my leaky blood vessels. See this little bunch of red that I see right here? Little bunch of red right here, and there's a little bit more down here. Each of the fluid-filled spaces in the brain, called the ventricles, each of them have choroid plexus. And remind me, we just talked about this. The choroid plexus of the brain, what is its job? What does choroid plexus do for your brain? Besides just leaking, right? It's leaking on purpose. Yes, it's leaking to make that cerebrospinal fluid. Absolutely. Okay, so check this out. We have 4 fluid-filled spaces in the brain. We will talk more about these on Thursday. We have 4 ventricles or fluid-filled spaces. Each of these 4 spaces has choroid plexus. All have choroid plexus, which means that when I'm making my cerebrospinal fluid, I make it in each of these fluid-filled spaces. If I'm constantly making my fluid, I constantly need to be moving it as well. So I build something and I got to press it on. It's kind of like an assembly line. So think of this choroid plexus up here. The first ventricles that you have in your brain are called the lateral ventricles. They're the 2 biggest ventricles that I find in the cerebral hemispheres. We have choroid plexus that I find in the lateral ventricles. They make cerebrospinal fluid that floats around in these little horseshoe-shaped bubbles, basically, that are filled with CSF. But when it's done floating around inside this bubble, it needs to leave and go down to the next place. So it travels through a little passageway here called the Interventricular Foramen. It's a little tube here that goes down into my next ventricle. The next ventricle in the brain is called the third ventricle. And this one lives inside the duck face. It lives inside the thalamus. Inside the third ventricle, I also see choroid plexus. So inside here, I'm also making new cerebrospinal fluid. So I've got the fluid that came from the lateral ventricles plus the new stuff that I made in the third ventricle. This floats around for a while, then it needs to leave, so there's not too much pressure. I go down through a structure down here, a little tube called the Cerebral Aqueduct. So the Cerebral Aqueduct takes fluid from what's called the third ventricle, that's here inside the thalamus, here inside the duck face, down into the fourth ventricle. The fourth ventricle lives between the pons on the front and the cerebellum on the back. And notice that the fourth ventricle also has this choroid plexus back here. It has more leaky blood vessels. So I can make cerebrospinal fluid in each of these places, which means I constantly need to be draining it as well. So there are some structures called apertures, and we don't really talk a lot about them in lecture or lab, but I'll mention them here for you. Excuse me. An aperture is a hole. So I have holes that I find in the middle of the fourth ventricle, and I have some holes that I find on the sides of the fourth ventricle. These apertures, these holes, allow me to completely spill out my cerebrospinal fluid into the place that last week we talked about cerebrospinal fluid living, which is the subarachnoid space. The subarachnoid space is where my fluid goes when it leaves the ventricles. When I'm done with it, I'll send it out to the subarachnoid space. Some of it, though, also goes down, we see here at the very bottom, something called the central canal. Next week in lab, we're going to be talking about the spinal cord more in depth. And one of the landmarks we have to find in the spinal cord is this thing called the central canal. It's just literally a tube in the very middle of the spinal cord where there's some cerebrospinal fluid. Because remember, cerebrospinal fluid has oxygen and it has glucose inside of it. So I have this tube where I put some oxygen and glucose-rich solution in the middle of the spinal cord. The rest of it, though, goes into the subarachnoid space that floats around the brain, helps the brain to have a cushion in case your head goes forward really quickly. What we're talking about in lab this week are new structures called arachnoid granulations. This is, I mentioned it last week in our office hours about lab. These are little spaces where I drain my cerebrospinal fluid, because if I'm constantly making cerebrospinal fluid, I constantly have to get rid of it as well. So I get rid of it into the space that we labeled on our model last week as what we called the dural venous sinus. Or if you want to call it by its blood vessel name, they call it the superior sagittal sinus. Because see, here's the thing, we told you that fluid has a circle of life thing going on, right? So you told me that in the choroid plexus, when there's fluid inside there, we call that plasma, when it's inside the blood vessels. And then I leak it out into the ventricle, and I call it cerebrospinal fluid. And then I leak it back into the dural venous sinus, and it becomes plasma again. So we constantly have a relatively stable amount of fluid in the ventricles and a relatively stable amount of fluid in your circulatory system, because I do this circle of life thing where it goes from one place to another. I'm going to be mean and make you type. I'll give you some time to type here. What's it called if I don't drain enough cerebrospinal fluid? What's it called if I make as much cerebrospinal fluid as normal, but I don't drain it? Yeah, so multiple of us are mentioning it. The name of what it's called when I have too much cerebrospinal fluid is what we see a picture of here. It's called hydrocephalus. Hydrocephalus. If you literally translate it, it means water on the brain. That's literally what it means. But it's not actually a water problem. Hydrocephalus is a problem with cerebrospinal fluid. It's a problem where I have too much cerebrospinal fluid. So if for some reason one of those passageways between the ventricles gets blocked, now suddenly I've got a ton of extra fluid in the lateral ventricles. That could lead to hydrocephalus. Or if my arachnoid granulations, where it's supposed to be draining that fluid, if they get clogged, I could develop hydrocephalus. So hydrocephalus is when I have too much cerebrospinal fluid, and the way that it presents itself is you typically see this great enlargement or the great swelling that I see here in the head. Now, one of the things that you learned about in the lesson with hydrocephalus is we said that hydrocephalus is dangerous in babies, and it is lethal or deadly in adults. Why is it, what can babies do that adults could not do? Or what do babies have that adults don't have that would allow them to adjust to this extra pressure? Absolutely. Remember way back on day one when I taught you guys about fontanelles, way back in the day. Here's why we talked about fontanelles. Hydrocephalus. Babies have fontanelles. Fontanelles are the soft spots that traditionally or primarily are used to help a baby be born. But if for some reason the baby has too much cerebrospinal fluid, that actually gives space for the skull to expand with the fluid. Now, to be fair, we're absolutely not going to leave it untreated like this. Eileen was mentioning a family member that had a baby with this condition. Yeah, you don't just let this condition go on forever. We don't just leave all this extra fluid here. You use what's called a shunt or a tube to take that fluid that we have accumulating up here in the ventricles or around the ventricles and drain it to other places in the body. But in babies, they have the luxury, if you will, of having these fontanelles. They have the ability to stretch or change the shape of their skull bones. What is it that adults have instead of fontanelles? What do adults have instead of fontanelles? Adults have, somebody mentioned it. Yeah, adults have sutures. Sutures are places where the skull bones come together and they don't move at all. So that's great for keeping your brain safe. There's no movement for your brain. But if we start to get this extra cerebrospinal fluid, if we get extra pressure up there, there's nowhere for that pressure to go except down onto your brain, or more specifically, onto your brainstem. So hydrocephalus is something that's very dangerous in toddlers and children and adults because it puts so much pressure on the brain. I don't know if there, so the thing with hydrocephalus, somebody asked about the lifespan of someone with hydrocephalus. If we treat it, if we identify it and treat it, you can live normally for a very long time. You just kind of have a tube that will run from somewhere here in the skull. Typically it runs down through the neck, down into the abdominal cavity, where it just literally drips fluid all the time. So as long as we diagnose it and as long as we treat it, you can have a relatively normal life. Earlier there was a question in the chat about what can block these things. A lot of times what's blocking it is either a tumor that develops or it is a malformation in a structure. So something that was always the wrong shape, or we could get a tumor that could be in its place. Symptoms happen quickly. Is there a time they have from start to death? That's a great question, Carrie. I don't know. How quickly from someone developing hydrocephalus to it killing them, I'm not sure how quick it is. That would be my concern when we talk about diagnosing hydrocephalus. Like, how quickly do we figure out that that's the problem? Is it fast enough for us to be able to get that shunt in place and save the person? I don't know the answer on that. But if it's kind of a slow growing tumor that's putting pressure or blocking that passageway, maybe if it develops slowly over time, we might have enough time to put that shunt in place. Okay, so hydrocephalus, if you are looking at your learning objectives for the lesson, and remember I told you that's still your best study guide for Unit 3 exam, you'll notice that the last learning objective for Lesson 11 lists a bunch of different disorders, and I want you to be able to explain what causes them. We just talked about what causes hydrocephalus. So this helps you to understand what causes the condition called hydrocephalus. We would say the brain structure that's malfunctioning in hydrocephalus would be those choroid plexuses, or we could say too that it's those ventricles, the structures I use to move the fluid through the brain. So hydrocephalus, an issue with the choroid plexuses and with the ventricle system where I send my fluid around. We've got choroid plexuses, and remind me, these ones did or did not leak. Were the choroid plexus blood vessels leaky or not leaky? Yes, they do leak. These are the leaky ones, absolutely. Okay, so the other kind of blood vessels, the main blood vessels that we have in the brain are the ones, again, that we talked about on day one, called continuous capillaries. Continuous capillaries. Most of the blood vessels in your brain are these continuous capillaries. So if you remember back from lesson number 1, continuous capillaries get their name because they have continuous layers of cells inside them. So if you see this yellow line here, that's all my cells that make up the wall of our blood vessel. They also have a continuous layer of connective tissue around them. I see that here in orange. So continuous, yeah, like Eileen mentioned for us, no holes. So a continuous capillary, there are no holes. There's no spaces for things to get out. Now, when we're in the brain, the other thing we do to keep ourselves extra safe is, in addition to having this layer of cells with no holes, and in addition to having this connective tissue that wraps around where there's no holes, we also actually add an extra layer of cells around this. Yeah, so Carrie's chiming in with us. Continuous capillaries are wrapped in cells called astrocytes. And we're going to want to underline, highlight, star those guys. The reason we want to underline, highlight, star these is, you know how Dr. Allis really likes compare and contrast, right? So it would be a really great idea. Here's a wink, wink, nudge, nudge study idea for you to make yourself a chart that says continuous capillaries on one side, and on the other side of the chart says choroid, if I could spell, choroid plexus. Both of these types of blood vessels wrap themselves up in cells, but they wrap themselves up in different cells. So one of the things that we could put on our chart is who wraps them up, what kind of cell wraps them up. We could put notes on our chart. Are they leaky or not? We could put some messages about what gets through. These are the kinds of things that we would want to compare and contrast on a continuous capillary versus a choroid plexus. So here's a wink, wink, nudge, nudge. I know you guys said that you missed those from class, right? So here's your wink, wink, nudge, nudge. These are the kind of compare and contrasts that we want to make between the blood vessels we just talked about, the choroid plexuses, and the ones we're going to talk about now, the continuous capillaries. So continuous capillaries, in general, we would say that these ones do or do not leak. Do we think continuous capillaries leak? These ones do not leak. Yeah, these are the ones that we said on day one are the ones that do not leak, hardly at all. The reason that they don't leak is because of those continuous cell layers, and also because we wrap them up in what we call astrocytes. Now, when we take these, I'm going to abbreviate here, continuous capillaries, and we wrap them up in astrocytes, we generate what's called, we'll do equals, the blood-brain barrier. The blood-brain barrier. That's some terminology you want to be familiar with. I'm sure you've heard of this before, the blood-brain barrier. And the blood-brain barrier, its job is to keep bad stuff that might be floating around in the blood vessel, keep it inside the blood vessel and not go into the brain tissue. Because the brain tissue doesn't have a lot of immune system support up there. It is what we call immunoprivileged, meaning there's not a lot of white blood cells that live inside the brain tissue. At least not normally. It's bad news if normal white blood cells have to go into your brain. That means we probably got a really bad infection. So the blood-brain barrier is the tissue inside my blood vessel plus those special cells I wrap around it. The blood-brain barrier, essentially, think of it as three plasma membranes. Three plasma membranes. We've got one from inside the cells that make up the blood vessel, one from those cells that help me to make fibers and connective tissues, and one from those astrocytes that are around here. I basically wrap up my blood in three plasma membranes. So when we talked about the plasma membrane of the cell, blast from the past here, what were some of the ways that we said we described the function of the plasma membrane of the cell? What's the function of a cell's plasma membrane? What does it do? Oh, I promise it's not a trick question. Think back on that cell classroom. Hey, now that we're like locked in our individual homes, is anyone interested in getting locked in a classroom together now, right? Now that we can't leave our houses. It keeps things from coming in and out. Yeah, no, we don't want to get locked in the classroom either, right? We just want to be freely moving, right? No plasma membranes, no walls. Yeah, so the job of the plasma membrane was to divide the inside and the outside, or it controls what gets in and out. It controls what gets in and out. When I ask you, because I did ask you on your lecture outline, and I did ask you, I think, on the homework assignment, and I will ask you on the test, what can and cannot get across the blood-brain barrier, this is the same question that I asked you way back in Unit 1 when I asked you what can and cannot get across the plasma membrane of a cell. Because the blood-brain barrier is just 3 plasma membranes. So if I'm a molecule that can get across 1 plasma membrane, I can get across 2 plasma membranes. I can get across 3. It's not a problem for me. So let's remind ourselves, when I asked you guys way back in Unit 1 to describe the kinds of molecules that can diffuse straight through the plasma membrane, what were some of the words that we used to say this is a molecule that goes straight through? Yeah, so the first word that we used, things that are nonpolar. Remember when we talked about the structure of the plasma membrane? Was it the inside or the outside of the plasma that decided what came in? Let me draw a really terrible plasma membrane here. Look at this awesomely terrible plasma membrane here. Yeah, as I'm drawing my terrible membrane, you guys are telling me it's this inside part of the plasma membrane that decides what comes in and out. This inside part in here, as several of you have noted, is nonpolar. So it's nonpolar. If you match me and you are nonpolar here in the middle, you can get through. And as somebody mentioned for me, there is a synonym for nonpolar, and that synonym that they mentioned for me was hydrophobic. I'm afraid of water. Hydrophobic. One other word that we could use as a synonym for all of these things, these little guys here in the middle, that my little terrible lines here, what are these called in the plasma membrane? Yeah, these little things Carrie's mentioning for us, Ariel's mentioning for me, these are lipids. These are my ugly little lipids here in the middle. So one other word that I could use that means the same thing as nonpolar and hydrophobic is lipid soluble. Lipid soluble. Yeah, they're the bouncers of my plasma membrane. They decide what gets in and out. Okay, all of these words right here, nonpolar, hydrophobic, lipid soluble. These describe the kinds of things that can cross the plasma membranes and therefore can cross the blood-brain barrier. So when we're looking at our picture here to figure out what can and can't cross the blood-brain barrier, one of the things that we see going straight across the blood-brain barrier here is carbon dioxide. And I know it's been a way long time since we did chemistry, but carbon dioxide is a molecule that does share its electrons equally. So it is considered nonpolar. Nonpolar things fit my list here. They can go straight across the blood-brain barrier. Carbon dioxide can go in and out of the blood vessels in the brain tissue very easily. Same thing down here with oxygen. Oxygen can go in and out really easily, no problem, no help required, because we are non-polar. We share our electrons equally. Notice up here higher, we have fat-soluble molecules. Fat-soluble is another word for lipid-soluble. Fat-soluble molecules, they can pass straight through as well. They go in and out as they please. When we start talking about things like glucose, when we start talking about things like amino acids, notice how for both of those, they include this word transport. Transport. When I use the word transport, I have to use a special tunnel to get things from inside the blood vessel out. Does anyone remember the kind of molecule that we said kind of makes us tunnels or makes a way for things that are polar to get across a membrane? What do we use to help move things across the membrane if they're not going to be able to pass through the membrane by themselves? Anyone have an idea? Yeah, my channels. Yep, channels are aided, or in big picture, what we're talking about are proteins. Try to draw a little protein channel for you right here. Here's my little protein channel. Looks like a blue hot dog. So we got going on here fat-soluble things. These go straight through the membrane. No big deal. But we start seeing this word transport. Transport means I needed some help. So for me to get glucose inside my cell, it's been a while since we talked about glucose, but I'll remind you, glucose is polar. So a polar molecule can't sneak through the membrane. I'm going to have to use a transport protein to be able to get glucose inside. Or I have to use transport to move amino acids. Now, probably not my non-polar amino acids, but my polar ones or my charged ones, they don't play nice with my lipid tails in a membrane. They're not going to play nice with the blood-brain barrier. So I have to use a transport protein to get these things across. Notice here too, you're seeing positive and negative. We got our little ions right here. Hey, terminology review. When I talk about a positively charged ion, what do we call those positively charged ions? Positively charged ions, that's the cation ions, exactly. Cation ions have a charge. They're not going to get through unless I make them a protein channel. Same thing with the little anions over here. Things that have a charge are not going to get the blood-brain barrier. And the last thing we see in our picture here are really large molecules. Things that are really big, it doesn't matter. It's a problem with the size. If you're super big, there's not enough space for me to squeeze between, because if we were technically accurate, right, all of these are little circles. I can't squeeze between things if I'm super huge. Plus, if I'm super huge, the chances of me having some part that is either polar, like glucose, or some part that has a charge is really high. Things that are really large can't get across the blood-brain barrier. So you've got some questions, some molecules listed there in your lecture packet to help you predict whether or not they can get across the membrane. Again, my big rule, the way that I want you to think about and study it, just think of the blood-brain barrier as 3 plasma membranes. Just like we did way back in Unit 1 when we predicted what can and cannot get across a plasma membrane, make those same kinds of predictions when you're talking about the blood-brain barrier, because it's the exact same kinds of things that can get in and out of the blood vessels. Thumbs up or questions? How do we feel about the blood-brain barrier? Got a smiley face? I'll take that smiley face. Okay, well, if you think of some more questions as you are reviewing this material, you're working on that homework assignment and getting ready for the exam, please feel free to email those, or again, tomorrow our session is just review. So we're going to talk about whatever you guys want to talk about. Bring your topics tomorrow so that we have stuff for us to talk about. I'm going to flash up one more time. For anybody who happens to not make it yesterday to office hours, I want to flash up the exam 3 details for you again, just so that they're there. Help me out. I think I had people from every class here yesterday. Did all of the group me's get these details sent out Monday, Wednesday, Tuesday, Thursday, and Friday? Okay, I know I got some Friday friends saying that. Okay, Tuesday, Thursday did. Monday, Wednesday, can we confirm for me? Yep, and Monday, Wednesday did too. Awesome, perfect. Okay, so just a reminder for you here on those details. Starting tomorrow morning at 7:30, that exam goes live. But again, I am going to do a review session with you tomorrow at 9:30. So I don't expect a lot of us to be working on the exam right away when it posts. Remember, when you sit down to take the exam, remember that once you open it, you are committed to doing it. So please do not open the exam. Please don't play with the exam until you're ready and committed to taking that exam. Because once you open it, your clock starts. You got 60 minutes to complete it. I'm not sure what math you're doing, Pilar. 45 out of 100. Yeah, asking about the group work. So Carrie, the group work deadline, and this might be another thing to make sure that group meet gets out in the group meet. I have extended the group work deadline to Friday night. So I would say wait until Thursday night to work on answering questions that your group members haven't started on. You might want to answer those questions yourself, just for your own reference, but don't start uploading your answers to those questions till maybe Thursday. Give your group mates a little bit more time. So it might be, Pilar's asking about the questions, what they're worth. Your percentage out of 100. Some questions might be worth more than 1 point if they have 2 parts to them, for example. So my guess would be maybe it's going to be closer to 50 points, 60 points maybe, but it's going to be a percentage. So you'll see a percentage score. Is it actually a long time per question? Eileen asks. It's longer than it would have been in class. Remember in class, I told you guys if we took this exam together in class, it would have 70 questions and you'd have 80 questions. So that, yes, you're getting longer than in class. Group work, does there need to be a reply by everyone or an edit? Everyone may not participate. Most people have been really good about still participating in our class and logging into Blackboard since we've transitioned to online. Some people have kind of disappeared a little bit. I will be grading the 2 ways I'm going to grade this assignment. Number 1, if you do not participate in it at all, you get a 0. Number 2, so if you participate at all, if you're a regular contributor to your group, then whatever score you're getting, if your group is getting, you are getting that. Number 2, I'm grading it for accuracy. So make sure at the end that the answers are accurate. Again, the end is Friday night. So there's still some time, hopefully, for group members to participate. I know some of us are mentioning that we emailed a while back and haven't heard from them. So if you haven't heard from your group members Thursday night, Friday, they haven't contributed, go ahead and answer those remaining questions. I know it sucks, but I can see how much people are involved in it. So I promise if you did all the work for your group, you will be the only one getting credit for that. So it doesn't have to be and has to contribute. You make sure the answers are correct. You get credit for the person should do well on the exam. I think you should. Yes, absolutely. Prepare for the exam just like prepare for class. And use those detectors to measure information. Feel free to keep checking out each group or if you want to excursions out, get in touch with her. Prepare if you for the exam, prepared for classics. I think you guys are do just fine. I'm in and out. Not on my showing it. That actually shows me. Can we hear me again? Am I back? Back now. Okay. So all I said is just prepare for this exam like you'd prepare for a normal lecture exam, and I think you're going to do just fine. So put in the kind of studying, the way that you studied. Make sure to give yourself a distraction-free environment to take the exam. I don't think you should freak out about it more than you'd freak out about a normal exam, which I know that we all freak out about normal exams too, but try not to lose your head, right? Like I always tell you, and try to go with your gut, because if you change an answer, you probably change it to wrong. No, there's probably not going to be a curve, and we're all, well, I won't say we won't all get a 100, because we might if we prepare correctly, but I believe in you guys. I really think that you guys can handle this. I believe you can do this. Pilar, there's no bonus questions on the exam. No, I'm still working on bonus activities for the course, but I don't want to overload you guys with assignments. So I have a couple of ideas of things that I think I might roll out sometime soon, but again, I don't want you to feel like I'm just giving you 10 million things to do all at once. All right, I am going to close out our recording for the day, and then I'll stick around for a few more minutes if you have last minute questions. But again, please plan to come tomorrow, 9:30 to 11:30. But again, I really need you guys to come with specific questions because I have no plans for tomorrow. So we'll get really bored if nobody brings any questions. So bring me your topics tomorrow at 9:30, and we'll do a review together. So goodbye to my recording friends. I'll see you again soon.